Okay, welcome to everyone joining us here. Today's talk is entitled Next Generation Muscle Tissue Engineering, iPSC-Based Approaches for Scalable and Reproducible 3D Models. My name is Ariana, and I will be helping guide us through this event. The webinar is designed to be interactive. So if you all would like to go ahead and let us know in the chat where in the world you're tuning in from right now, that would be a great way to start. Additionally, the webinar does include a Q&A session. So please feel free to submit questions or comments for our speaker throughout the presentation. For those unfamiliar, you can find the Q&A and chat functions at the bottom of your Zoom screen. We'll be attending to those Q&A questions after our main presentation. Now, it is my pleasure to introduce our speaker for this event. Dr. Shawn Luttrell is the product manager of cell and tissue models here at Curi Bio. Just to give us a little bit of background, Shawn graduated from the University of Washington with a degree in biochemistry and molecular biology, followed by graduate studies in developmental biology. She completed a postdoctoral fellowship at the Institute for Stem Cell and Regenerative Medicine at the University of Washington, where her focus was on stem cell and gene therapy treatments for neuromuscular diseases, with an emphasis on iPSC-derived muscle development. Currently at Curi Bio, Shawn leverages that expertise and plays a pivotal role in the development of Curi Bio's iPSC-derived cell lines and tissue models to increase the preclinical utility of in vitro cell-based assays. Okay, without further ado, I'd like to hand over the presentation to our speaker. Shawn, please begin whenever you are ready.
Okay, great. Hello, everyone. Can you all hear me and see my screen? Yes? Yes, okay, great. Thank you for joining our webinar bright and early on this beautiful morning in Seattle, Washington. So I actually see we have attendees from all over the world, though. So I wish you all a good day or a good evening, wherever you are joining from. So my name is Shawn Luttrell, and I'm product manager for cell and tissue models at Curi Bio. And thank you for that lovely introduction, Ari. Today I'll be talking about some of the technologies and cellular models we've developed here at Curi Bio, aimed primarily at improving the physiological relevance of preclinical in vitro human tissue models. So I'll show you some of the tools we have designed for 3D muscle tissue engineering to improve the reproducibility of fabricating muscle organoids, and also importantly, the ease of making these tissues. So we use induced pluripotent stem cell derived cells in a variety of different contexts to make bench work more translatable to model development. Yes.
Sorry to interrupt. I actually can't see your screen. I can just see you.
Okay. Looks like we are having some technical difficulties, so let's back up for just a minute. Okay.
There we go. Perfect.
Okay, sorry about that, everyone. Let's try this again. So I don't think we need to restart the introductions, but I will restart on kind of what this talk is going to be about. Basically, I'll be sharing some information on new iPSC-derived model of Duchenne muscular dystrophy that we just released commercially. And we've specifically optimized this line for 3D muscle tissue engineering. And I'll show you how to make these tissues using our platform and the different kinds of data you can generate using our cells and biosystems. With that, as I mentioned, we are here in Seattle, Washington. We're a biotech company based out of UW, actually. You can see beautiful Mount Rainier there in the background, and our headquarters are located down on the water in the Belltown area in beautiful Elliott Bay. So at Curi Bio, we integrate biology and engineering into a preclinical pipeline. And we do this with 3 sets of technologies and tools. So human cells derived from primary and induced pluripotent stem cells, proprietary biosystems that we use to measure functional phenotypes from these cells and tissues, and then AI and machine learning to generate meaningful data from these biosystems. So in the last several years, we've been primarily focused on contractile and excitable tissues, so skeletal and cardiac muscle. But more recently, we've been moving into the motor neuron and NMJ space. And our goal, of course, is to create human in vitro models and platforms that better replicate in vivo biology to improve the translatability of preclinical testing, with an end goal of more accurately predicting human responses to compound exposure to expedite the discovery of life-saving therapeutics. So on the bottom of these contractility traces on the right, you can see one of our 3D engineered muscle tissues. And when we look at the force frequency waveform that's measured on our man-array contractility platform, you can see that this trace is nearly identical to in vivo muscle assessments across several different model systems, including humans. And that's really one of our goals here at Curi, is to create these models and biosystems that replicate this in vivo biology. And that's one of the advantages of 3D culture systems. So before I came to Curi Bio, about 3 years ago, I was doing a lot of work making induced pluripotent stem cell lines and then optimizing protocols and media to differentiate those cells into skeletal muscle. A large part of that work was culturing the muscle in 2D to assess development and function. And 2D culture is great for a lot of different assays, but long-term muscle development to drive maturity in iPSC-derived myotubes is certainly not one of them. So coming to Curi Bio and working in 3D was really a game changer for me, because now we can keep these cardiac and skeletal muscle tissues in culture for months and months with our proprietary media. And anyone who has cultured myotubes in 2D knows that they will likely delaminate from the substrate as soon as they develop and start forming that passive tension and those spontaneous contractions. So modeling development and maturity in 2D is quite difficult. So we can co-culture different cell types together to better mimic the in vivo development and also disease phenotypes that might only present when certain cell types are cultured together. We can also alter the ECM in 3D systems to better replicate in vivo architecture and complex cell ECM interactions. We can measure muscle function through contractility over time and use our biosystems to stimulate these tissues, modeling exercise, inducing maturation, or even injuring these tissues to model regeneration or damage prevention after applying recovery compounds to these tissues. So while 2D systems are good for their simplicity and throughput, 3D systems are really vital to better replicate in vivo biology and function. So at Curi, we talk about having a discovery engine. So different technologies and an overall integrated process. So we work with our partners and customers in a variety of different ways, starting at the top with sourcing our cell lines. So we do make many of our iPSC lines in-house that represent a wide range of cardiac and neuromuscular diseases. We can engineer mutations into healthy cell lines, or we can correct patient mutations to make isogenic cell models. We differentiate our iPSCs into different lineages, including skeletal muscle and motor neurons. And we actually obtain our iPSC-derived cardiomyocytes from our affiliate company, Celogics. And Celogics can actually differentiate any of your iPSC lines into cardiomyocytes if needed. You can find them on our website or on their own website. But then we put our differentiated cells into our 2D and 3D tissue models to measure function in a variety of different ways across our different biosystems. So we can treat these muscle tissues with gene therapy constructs or drugs or antibodies or AONs or whatever your therapeutic is, and then screen those models for therapeutic rescue or run safety talks or potency assays. This data is then fed into our automated analysis tools to get results in a more high throughput approach. So you can see we work very flexibly across many disciplines, but we're mainly focused on safety toxicity, disease modeling, and therapeutic discovery. So for the rest of this talk today, I'm going to focus on our DMD model cells and our biosystems that we produce to streamline 3D tissue engineering and data analysis. So here's a look at some of our biosystems designed for 2D and 3D applications. So with our platforms, we're really trying to drive maturity in vitro tissue models, as well as focusing on measuring functional aspects of the cellular and tissue level physiology and throughput of these models. So the first product we actually brought to market at Curi Bio was our line of nanosurface plates on the top left. These engineered plates provide a biomimetic scaffold that directs the structural development of 2D monolayer syncytial tissues like cardiomyocytes to more accurately recapitulate the structure of the native myocardium. So these plates also provide alignment cues for skeletal muscle and other types of cells that require that organized alignment to develop properly. So a second platform that we use to create these in vitro models is our cytostretcher in the top right. This instrumentation line is focused on providing biomechanical strain to in vitro cultures, both in 2D and in 3D. And our 3D model is actually nearly ready for beta testing. The Nautilus is our optical mapping system that can be used with both 2D and 3D tissues as well. And this is where we measure our voltage and calcium flux in our muscle tissues and cardiomyocytes and motor neuron cultures as well. And then finally today, I'll be talking about our ManoRay platform. This is our 3D contractility measurement stimulation biosystem. This is a newer product for us, and we're very excited about it in terms of improving the throughput of contractility measurements in vitro. So before I move on, talking about our instrument and our cell models and our 3D tissue engineering, we're going to take a quick poll to see what your research interests are, and so I can try to tailor the rest of my talk to fit the interests of the group. So you should see a poll up on your screen asking, what are your application research areas of interest? And, please select everything that applies to you. And we'll just take 15, 30 seconds or so to gauge the audience. So it looks like you're all, spanned out over these areas, but a large percentage of you are interested in cardiac, muscle, functional output, contractility, disease modeling. That's wonderful. That's exactly what this talk is focused on. So perfect. Yes. Looks like the audience is in line with the direction that this talk will be heading. So let's move on into our biosystems. Great. So thank you for taking that quick poll. Sorry about that. Let me get rid of that. There we go. So this is our ManoRay instrument on the left here. But before I dive too deeply into the platform, I want to talk about our engineered muscle tissues and how we make them. Then I'll explain the instrument, how it works and what it does. And then I'll talk about the kinds of data that you can get from this platform using our cells and media. So this is an engineered heart tissue. So this is a video of one of our constructs. Skeletal muscle is going to look almost identical to this. So cardiac tissue will start to spontaneously beat usually 2 to 4 days after casting the tissue. This is one well of a 24-well plate, and we're looking up through the bottom of the plate. So we use a 2-post system, and we have a rigid post on the right here. Get my pointer working. And well, we'll forget the pointer for now. We have a flexible post on the left, and every time this tissue contracts, that flexible post will move. And you'll notice, I'll draw your attention to the small black square inside that flexible post. And that's actually a high-powered magnet. So every time the flexible post moves in response to muscle contraction, the magnet moves. And that's essentially how we measure contractility in our instrument. And I'll talk more about that in a few slides. So these tissues have been around for a long time. We're certainly not the first to make them, but Typically, making these constructs has been a very painstaking process. Additionally, making reproducible and consistent tissues every time is very challenging as well. So the first thing that we really wanted to tackle at Curi Bio was to engineer a device for 3D tissue casting to improve the reproducibility, consistency, and also just the ease of generating these constructs. So with that, we designed a 24-well consumable casting plate. This plate has a top lattice that holds 24 sets of posts, one set for each well, and then a bottom casting tray. Inside each well of the casting tray is a trough, and there's a blow up of the trough on the right side here. And this is where you're going to put your cell hydrogel mixture. And essentially, it's like making a popsicle. You add your lattice into these molds containing your cell suspension, transfer the plate into an incubator for 80 minutes. The hydrogel will compact around those posts, and then you transfer these tissues to a new plate of media, and you're up and running. You'll transfer the lattice with all of the tissues to a new plate of media every 2 to 3 days, and really that's all there is to it. So it really is that easy to make these tissues, but does this consumable actually improve the reproducibility and consistency between tissues and between batches of tissues? And so here's some data across 4 plates of tissues looking at twitch force, the muscle constructs as they develop over the first 3 weeks of culture. We also have compaction of the tissues, measuring the diameter of the tissue in the midsection over that same time frame. And you can see by these error bars that there's actually very little difference in these metrics between the tissues in the same plate, but then also between plates of tissues. So it does seem that this consumable does improve, excuse me, the reproducibility and ease of making the tissues. So then the next thing that we wanted to look at Curi Bio was how do we measure function in these tissues in a more high throughput way? Excuse me. And so typically function in these 2-post tissues has been measured using optical methods. And this is a diagram I pulled from the literature that shows what someone might have to do to measure these optically. So you would capture a video of the tissue beating and then measure the displacement between the relaxed state and the contracted state. And then using pixel to micron conversion and some fancy math, you can then convert that into a contractile force in micronewtons. The problem with optical systems is not only the complexity. So here we have a microscope, a stimulator, a data analytics program. You're probably going to have to know some sort of programming to calculate your forces, perfusion equipment. But also another big drawback is just the throughput. So usually you're measuring one tissue at a time in an optical system. And that's really not conducive to any kind of high throughput drug studies. And so to tackle that, we engineered our manta-ray instrument, and that's pictured here on the left. And that's one of the real advantages of the manta-ray is we can measure contractility across 24 tissues simultaneously with basically a push of a button. So this instrument is about the size of a shoebox, and it will fit right inside of a standard cell culture incubator. The instrument essentially acts like a plate reader. And so it has an array of magnetic sensors that are right below that plate. And that will detect movement of that flexible post in every well simultaneously. So furthermore, our engineers have designed a custom stimulation lid, well, for all tissues, but specifically for skeletal muscle, since it doesn't spontaneously beat like cardiac muscle, we need to stimulate it to force it to contract. But you can also pace your cardiac constructs or your motor neurons or your NMJs in our NMJ platform that's in development. So this will work across many different cell types and systems. So the stimulation lid, there's a top-down view in the middle, and then there's a side view right below that. And the stimulation lid will fit right on top of that lattice. And so you end up with 2 sets of carbon electrodes that are situated on either side of the tissue. And then you can control stimulation through the software to stimulate one tissue at a time, or you can stimulate all 24 tissues if you want. Oops. Get that to work. But then as you stimulate these tissues, that flexible post will move, and then that's converted into these contractile waveforms that I'll show you more of that data here in just a minute. So we can run simple twitch stimulations to pace these tissues at 1 hertz, or we can increase the frequency of stimulation to drive these tissues into a fused tetanus. It's really whatever muscle output you're looking for. So this is, oh, there we go. It's working. Sorry, having a couple of technical difficulties here. So this is what the software looks like that pairs with our ManoRay instrument. And again, we're focused on simplicity and flexibility. So here you can see contractility traces from 6 tissues in the upper left quadrant of the plate. You can look at 6 tissues in live view at a time. But when you do a recording of these tissues, you're recording data across the whole entire 24-well plate at once, simultaneously. So here in the bottom left is what a full plate of these engineered muscle tissues looks like. When we go to our stimulation studio feature in the software, this is where you're going to design your stimulation programs. So you can design using different pulses, so a biphasic or a monophasic pulse, and you'll just drag those. Sorry, I just can't get my pointer to work here, so I'm going to have to describe this in words. So you can drag these waveforms down to your protocol bar where you're designing your stimulation protocol. And you can design simple twitch, stimulations, or really complex, force frequency protocols. And then assign those protocols to any well in the plate. So you can stimulate 1 well if you want, you can stimulate a column, you can stimulate a row, or the entire plate. You can use 24 different stimulation protocols if you wanted. It's really whatever you design to fit your research needs. So as I said, when you take a recording of these tissues, that data goes up to the cloud if you want. And you can get back a few minutes later an Excel sheet with all of your contractile metrics from each tissue, fully tabulated. Here I'm going to play a little video that walks you through this process. So here we're going to live view, and you're going to see again the live view of 6 wells at a time. But again, we're recording all 24. The colored bar on the left indicates the stimulation has started in those 2 wells. We can see force on the X and time on the Y. So you can see in these 6 wells, we have different contractile forces for these 6 tissues that are all pacing at about 1 hertz or so. So as I said, when you take a recording of your tissues, that data will go up to the cloud. And then just a few minutes later, you're going to get an Excel sheet back with all of your contractile metrics for that particular waveform for every well. You get force as well as kinetics across the entire contraction. So you can take a 30-minute or a 30-second recording or a 3-minute recording, however long you want, but you'll get that data analyzed back in a matter of minutes. And this is really one of the major benefits of our ManoRay platform and cloud analysis software tool, which is called Pulse 3D. So using an optical system to measure function in 3D tissues is laborious. It can take days to fully analyze all of these metrics. So with Pulse 3D, you take a recording of your tissues, and then you get that data back fully analyzed in this Excel sheet where you can pull that information out, plug it into your graphing software, GraphPad or whatever you're using, and you basically have your data ready to present at your next meeting. So if you want, one of the other features of our cloud analysis software, Pulse 3D, is there's an interactive analysis feature that lets you customize some of these metrics to fit your needs. So let's say you're interested in time from peak to relaxation 30. You can tell the software to analyze that region of your waveform, and then it will reanalyze that data and give you an Excel sheet back with that new metric in there, in addition to all of these other metrics, looking at force and kinetics across the contraction waveform for all of your wells that have tissues. So what do these waveforms look like? What do the waveforms look like? So this is actually pulled directly from the Excel sheet. So on the top, we've stimulated the tissues using a 1 Hertz twitch stimulation. And you can see these tissues are contracting and they're relaxing, very rhythmically. They're, responding to being paced as we would expect. We have force on the X and time, or we have force on the Y and time on the X. So we can increase that frequency of stimulation and we can drive our skeletal muscle tissues into a fused tetanus. So here we see a semi-force frequency relationship here where we're increasing frequency 10, 20, 30 Hertz, and we see a corresponding increase in force. I'm going to show you a better force frequency waveform that we've produced in our skeletal muscle tissues in a few slides. But before we dive into, the kinds of data that we can get from this platform using our DMD cell model, I'd like to start with a brief primer on Duchenne muscular dystrophy, because that's the cell model that I'll be talking about today. And so for members of the audience who are less familiar with this disease, I'm going to show you just a little bit of data on DMD. So Duchenne muscular dystrophy is the most common form of muscular dystrophy. Its inheritance is X-linked, so that's why only boys are affected. So DMD is a devastating progressive muscle disease that affects cardiac and skeletal muscle. Symptoms start in early childhood with difficulties walking, running, climbing. As the disease progresses, children are usually wheelchair bound by age 12. So few patients survive into their 20s. Respiratory failure was the main cause of mortality in these patients. However, with improved ventilatory support, cardiomyopathy is now the leading cause of death. So there is no cure for this disease. So there's an urgent need to pursue new strategies to prevent, halt, or reverse disease progression in DMD patients. So as far as the genetics go, DMD is a fairly rare muscle disorder. It affects about 1 in 3,500 male births. It's caused by mutations in the dystrophin gene, and the dystrophin gene is the largest gene in the genome. It is including exons and very large introns. It spans about 2.2 megabases. It's encoded by 79 exons, and it's translated into a 427 kilodalton protein. Mutations in this gene are usually deletions or duplications. And because the gene is so large, there are multiple, just a multitude of different kinds of mutations that cause DMD, and also a milder version of DMD called Becker muscular dystrophy. And so dystrophin, the gene dystrophin and the protein. So this is a protein that basically acts, it links the contractile elements of the cell to the membrane. So it acts as a shock absorber, if you will, between the cell's cytoskeleton and the extracellular matrix, protecting the muscle sarcolemma from damage during muscle contraction. So it stabilizes the muscle cell membrane by cross-linking the F-actin, cytosolic F-actin with the extracellular matrix through the dystroglycan complex. And then another critical function of dystrophin is to recruit signaling proteins like centrophins, dystrobrevin, NOS, so, and other signaling molecules that are really critical for muscle function. So one way to model development and treatments for this disease in vitro is by using induced pluripotent stem cells. And that is where our DMD model comes into play. So we have cells from sick patients with DMD that we've programmed into iPSCs to model different patient mutations. But then we also have iPSCs from healthy individuals that we've engineered mutations into the dystrophin gene to create our isogenic model of the disease. And that's the model that I'll be talking about today. So once we make the iPSCs, then we use a directed differentiation approach to generate our myoblasts after about 30 days. We enrich these cells using FACS to increase our myoblast purity. And then we expand our enriched cells to bank for quality control. After these cells pass our internal QC metrics, we can use our proprietary medium to differentiate the myoblasts into myotubes in 2D on our nanosurface plates. So in the bottom left, you can see an image of that, and you can clearly see that ordered arrangement of the myotubes on the plates that's driven by that nanoscale topographical surface. And you see that alpha-actinin staining some very well-developed sarcomeres in these cultures. Or we can put these into our 3D tissues. And the DMD line has been engineered with a deletion of exon 45 and a 17 base pair deletion in exon 54, leading to a premature stop codon to ensure this line is a true dystrophin null. So the first stop in our QC lineup is myoblast purity. So we want to know that the cells in our culture are at least above 80%. That's the threshold that we've set for 3D tissue engineering to achieve consistency between different cell lines. So the way we do this is to seed our myoblasts in low density and then fix and stain them with DAPI and Desmin, which is a muscle-specific marker. And then we can count our total nuclei per Desmin positive nuclei across 5 to 6 areas of the well to get our percent myoblast in the population. We just recently optimized a protocol to quantify purity using flow, which is much less laborious and likely more accurate since we're assessing every cell in the population. So we're transitioning to that method, but here you can see our purity is quite high after passaging these cells for expansion. 86.3% Desmin positive for our wild type line, and then 94.4 for our DMD line. And so we, as I said, we want to make sure these lines are comparable in terms of their myoblast percentage within these populations, because when we make our 3D tissues, we want to be confident that we're comparing relatively the same number of myoblasts between this isogenic pair of cell lines. So the next thing we want to test in our QC lineup is cell viability out of thaw, and then also cell fusion. So here on the left, you can see the general morphology of the myoblasts in 2D right before passaging. These cells are quite robust, and they have a viability range between 92 and 98% out of cryopreservation. And this is really one reason that we can put these cells directly into our 3D tissues out of thaw without any 2D culture or expansion. So we can be fairly confident that when we put these into our 3D tissues, most of these cells are going to survive. So the next thing that we look at is cell fusion in 2D. So it's not enough to know that we have myoblasts, but we want to make sure those myoblasts are actually going to fuse to form these multinucleated myotubes. And this is really more of a qualitative measure, since quantifying a fusion index is nearly impossible in my experience. But here we want to see basically a monolayer of contractile myotubes after just a few days in our differentiation medium. And that's exactly what we see. At this point, these myotubes actually start to spontaneously contract, and along with that passive tension that they've developed attached to this rigid substrate, they will basically rip themselves right off the plate. And that's one of the downfalls of these 2D cultures, and one of the reasons that we focus on 3D culture of our muscle constructs. So then after we confirm our viability, our purity, our fusion, then we actually want to validate these cells in our 3D system. And so with this disease model, we can see a clear disease phenotype that's presenting almost from the start of 3D culture. So these skeletal muscle tissues will be contractile after just a few days. So by 5 days, they're contracting under stimulation in the man-ray platform. And you can also see those spontaneous contractions that are strong enough to actually bend that flexible post just by eye. Usually happens right at that media change. But these are tissues from day 10. And if you look at the morphology of the tissue at the top, there's really not much difference that you can tell by eye between the wild type and the DMD line. But when you drill down a little deeper and look at the function and the microscopic arrangement of the tissue, that's where we can really see this pretty clear disease phenotype, both the force. So we're seeing probably a 3 to 4-fold difference in contractile force between these tissues, but then also the kinetics. So when we look at our contraction and relaxation velocity, our time to peak and our time to relaxation, we can see a very clear statistical difference between the two lines. And, two things about this. This is really important because it gives us a lot of different metrics, a lot of different ways to assess different compounds that might be aimed at treating this disease phenotype, to assess muscle function in different ways. And it's a testament really to the Man Ray platform itself, and just the wide array of metrics that we can get from this instrument. So this is a snapshot of just one day. So what happens to these tissues over time? So here we've tracked function in these tissues over 43 days. And we actually have an experiment going on right now that's been in culture for over 60 days using these cell lines. And even though the forces are lower, they're still quite functional. So they really last for quite a while to give you a pretty large experimental window to run your analyses with your different compounds, either for disease modeling or for therapeutic testing. So if we look at the morphology, we see that the tissues do get darker over time. And this is really indicating a greater level of compaction of the hydrogel. And this happens as the cells remodel the matrix over time. We include 10% fibroblasts in our cultures. So those fibroblasts are likely starting to lay down their own matrix. And they also help stabilize the tissues in these extended culture times. They provide, attachment points for our myotubes in our 3D culture to stabilize the muscle as it's contracting and developing. So if we look at the twitch force of the muscle from day 5, we see the tissues are quite strong early in development, and then they reach a fairly stable homeostasis, after 10 or 12 days or so, and then they're stratified for the most part between the wild type and the DMD line, with the wild type being significantly stronger for the first, over 35 days or so. So again, providing that long experimental window for you to test your different therapeutics. So one of the hallmarks of DMD is fatigue. So this is a great example of a clinically relevant aspect of the disease model. So when we stimulate the muscle, here we're using the twitch stimulation, you can see that the last contraction is producing less force than the initial contraction. So if we plot each of these force values as a percent of the initial force, we can measure our fatigue rate. And you can see that our DMD line is fatiguing much faster than our wild type line. So then you can start to ask questions like, does this drug improve the fatigability of the muscle? And you can couple this with other measurement techniques to determine damage, for example. So this just gives us another clinically relevant phenotype that we can use to measure any kind of functional rescue of different therapeutics. So the Man Ray platform enables us to assess the muscle in very complex ways. Aside from just a simple twitch and tetanic stimulation, we can really design these stimulation programs that test the ability of the muscle to move calcium and contract and relax appropriately in response to the type of stimulation. So one of the goals we have is to produce in vitro models that accurately replicate in vivo biology. And I love this slide because it so beautifully shows us how our in vitro data is a mirror image of in vivo muscle function. We ran a force frequency in these tissues, and as we increase the frequency of stimulation, we see the muscle moves from a discrete twitch contraction initially at 1 hertz, up through summation, 3, 5, 10 hertz, all the way into a fully fused tetanus at 30, 40, 50 hertz. Furthermore, we see a significant difference between our disease model pair that looks strikingly similar to the healthy and MDX mouse model of Duchenne muscular dystrophy right below that. If we plot each of these force values over these different frequencies, we can get a graph that clearly shows us the statistical difference in function between this isogenic cell model. So again, another way to assess the muscle, assess function, and then also assess recovery of different kinds of therapeutics. So we can fix these tissues at any point during 3D culture and run histology on them. So here we've embedded the tissues in paraffin and we've made thin sections for staining. So this is looking at a cross section of the tissue. At low magnification on the left, you can see that our myosin heavy chain positive myotubes in green are fairly uniformly distributed throughout the construct. So we're not seeing a necrotic core. Our tissue size is compatible with oxygen and nutrient diffusion throughout the construct. At high magnification, we can see a beautiful dystrophin ring that's localized to the membrane surrounding our pan-myosin heavy chain signal in green. So this tells us that our proteins are localizing to the areas that they should be, giving us more confidence that what we've actually made are contractile myotubes that are arranged as we would expect them to be in vivo. So even though we're not getting that nice syncytium of that highly ordered, tight syncytium of muscle tissue that you would see in vivo, the arrangement of these myotubes is fairly uniform throughout these constructs. And then importantly, dystrophin is absent in our DMD line. So the other thing that we want to test is, how do these tissues respond to drugs and different compounds? Do they respond the way that we would expect them to? So we put BDM on our skeletal muscle tissues, and this is a ATPase inhibitor, myosin ATPase inhibitor. So when we put it on our constructs, it should decrease muscle force, and that's exactly what we see in this dose-dependent manner. Importantly, when we wash this drug out, we see a return of function in our tissues. So we're not just simply killing the myotubes, we're actually, inhibiting contraction as the drug is intended to do. We can increase the number of concentrations that we apply to these tissues to get a full dose response curve with an IC50 value. So we can shut the muscle down. Can we increase force in the tissues? And yes, we can. So tirasemtiv is a troponin activator. So we should see an increase in contractile force when we apply this drug. And this was just a small pilot study looking at a couple of different concentrations. But you do see a modest increase in force with these two concentrations. And then again, a return back to that baseline function or near baseline function when we wash the drug out. So these tissues do seem to respond appropriately to these different compounds. One thing that we learned in developing our 3D skeletal muscle model was media matters. So what you feed your 3D tissues makes a big difference in the contractile function of the tissues. And when I first started working on our skeletal muscle model, I was using primary muscle myoblasts and what I would call a conventional medium. So this is a differentiation medium described in the literature for decades, consisting of high glucose and serum starvation with 2% horse serum and 10 nanograms per mil IGF-1. And the tissues, when I would use this medium, they were functional. They would fuse and they would form, contractile tissues, but the forces were really low and they didn't last. And that's under this longevity graph here at the bottom. You can see in green the conventional medium tissues would just crash after about 10 days or so. Before I came to Curi, I was working on different media. I was making these stem cell lines and then optimizing protocols to differentiate these into skeletal muscle. And part of that was to work on optimizing a media for the tissues. So I tried that. And sure enough, not only did the tissues, last much longer, but they were also stronger. They had better, faster kinetics. So our contraction and relaxation velocities are faster. And sometimes that, the slower contractile function can indicate impaired function. So there's something about that media that just isn't quite giving the cells what they need to develop properly and also maintain a homeostasis in these 3D tissues over time. And so with our Curi Bio medium, so we see this nice increase in force, and these tissues will last well over 2 months in 3D culture. And what we haven't done is really just taken them out indefinitely. So cardiac tissues, they'll last for basically as long as you can keep them sterile. Skeletal muscle tissues, we haven't just let them go long-term to see actually how long do they last. So do they reach this level of function and stay there indefinitely? We don't know, but they're certainly, functional for well over 2 months, giving you a very long experimental window to test your different therapeutics or disease model. So we tried this media on our iPSC-derived tissues, on our DMD model specifically, and we see very similar results. So if we just look at the pictures on the right, you can clearly see a difference in compaction of the tissues. Now, a lot of different things affect compaction of these 3D tissues, but remember, we put fibroblasts into our tissues. So it's very important to have a media that's designed to support multiple cell types over long-term culture. And we want to keep our cell numbers fairly consistent over time in these tissues, maintaining the overall health of the construct, while limiting significant proliferation of one or both cell types. So the level of compaction will also impact the force and kinetics of the tissue. And so we can see on day 8, tissues grown in conventional medium are basically non-functional. While tissues grown in the Curi biomedium, they're not only contracting faster, but they're also much, much stronger. So having a medium that's designed to support both cell types is really critical to achieve optimal tissue development and homeostasis over time. And so with our cell lines, with our DMD model cells, we actually provide this media to you. So you can buy our cells in different formats. So we have a 1 million, a 6 million, and a 12 million cell cryovial size, for 1 million being used mainly for 2D applications, and then the 6 and the 12 for 3D tissue engineering. So you can take these cells, thaw them, and put them straight into 3D tissues with our protocols. Using our consumables, and you'll be up and running and have contractile tissues just a few days after you make them. So with our cells, we provide up to 10 days of culture medium. So this is going to get you through that early developmental window, and you're going to have your strongest tissues after about 10 days. And from there, you can either end your experiment, keep them going out to 12 days or so, or you can purchase our maintenance media and keep these going for as long as you want them to last, certainly out to a couple of months. This media actually will last. Let me go back to this and mention one other thing. So you can use this media. I've tried it across many different cell lines. So it will work on primary cells. We have formulas for primary cells. They're slightly different between the iPSC and the primary line media lines. So you can use them on any cell line. And if you are making these 3D tissues and you're struggling with force and longevity, you might consider trying our media to see if that alone can make a difference, because it certainly did in my experience. So with that, I'll say, Man Array is really a sandbox for us as we're continually developing new tools and technologies to use with this instrument. So we now have an instrument called our 3D cytostretcher, our CS3D. And so this is really to model eccentric contractions and injury of these muscle constructs. So here you can see in the top right, this tissue is contracting. Now it's being stretched beyond its relaxed resting state. And we're doing this under load. So we're stimulating the tissue as it's being stretched to really get that eccentric modeling and injury modeling accurately recapitulated. Below that, we have our Nautilus system. So we can take our 3D tissues, we can load them with calcium indicator dyes. So in this case, they're loaded with fluorophore. And we can see every time we stimulate this tissue to contract that fluorophore will enter the cell. Calcium enters the cell, which is labeled with fluorophore, and we can see it light up. And then as the tissue relaxes, it leaves the cell. So we can measure that calcium flux using our Nautilus instrument. We can also do voltage mapping in this instrument on our cardiac constructs and neuronal constructs as well. We can load these tissues with AAVs, lentis, AONs, antibodies to deliver therapeutics at any time during development in 3D. So we can target whatever developmental window your therapeutic needs to address or needs to be applied at. We can do it from day 0. We can do it all the way at day 30. So if you have something that really needs to wait until the tissues are mature to address a phenotype later, we can certainly do that. And we're looking to partner with our customers to develop potency assays using our in vitro biosystems. We're currently working on incorporating motor neurons into our constructs to make neuromuscular junction models. And we're always looking for ways to expand our technology into other areas like lung, smooth muscle, cancer biology. So if any of those are interest to you, please reach out to us and maybe we can establish some sort of collaboration or partnership in developing these different platforms and model systems. So with that, I think I'll end there and summarize briefly by saying Curi Bio is very much focused on improving the preclinical development process for new life-saving therapeutics by humanizing the models that we use. We can benefit from the powerful technologies around human-induced pluripotent stem cell-derived cells, which can, of course, be differentiated into cardiomyocytes, skeletal muscle myoblasts, motor neurons, and a wide range of other terminally differentiated cell types. To improve the preclinical translatability of our toxicity, safety, and efficacy studies. So overall, we have a vision to reduce the reliance on animal testing using these technologies and ultimately accelerate market production of life-saving therapeutics while eliminating unsafe projects early on. So with that, I will just thank you for your attention and joining our webinar today. And if you have any questions or you want to learn more about any of our models or partnerships, you can reach out to me, of course, anytime, or Greg Lorman is our VP of Research and Partnerships. And Hee-Jun Choi is our Director of Sales and Marketing. So any of us can help you with any of your questions or get you to the people that you need to talk to get your experiments up and running. So with that, thank you. And I think we have time for some questions.
Fantastic. Thank you so much, Shawn, for that wonderful presentation. That does bring us to our Q&A portion. Everyone can continue submitting questions, but we have received a few already, so I will start with those. First question is, what kinds of cells have been validated using this platform?
That's a good question. So yeah, we've used, certainly skeletal muscle myoblasts, cardiomyocytes. We have an NMJ model that we're working on. It's a slightly modified consumable, taking a more compartmentalized approach using motor neurons and skeletal muscle myoblasts. Within those lineages, we've used a variety of different disease models. So we've looked at, DM1, FSHD, DMD, XLNTM, hypertrophic cardiomyopathy, dilated cardiomyopathy, BAG3. So just a variety of different disease models across those different lineages, so cardiac and skeletal muscle. So I would say our platform is really cell agnostic. You can probably use it with any kind of cell. So right now we have a strong interest in incorporating endothelial cells into our skeletal muscle and cardiac constructs to try to get some vasculature going on. And we have interest in quite a few other cell lines as well. So I would say, it's really compatible with just about any cell type if contractility is a metric that is an output that is, conducive to that cell type.
Wonderful, thank you. Next question, what other diseases have you been able to model using 3D EMTs?
Oh yeah. So I just went over, some of those, but basically we've looked at a wide range of different disease models, and our customers and partners have also looked at various diseases in both the cardiac space and the skeletal muscle space. So on the skeletal muscle side, what we have internally is DM1, FSHD, DMD, and XLNTM. And within those disease models, we have isogenic pairs, and then we also have patient lines looking at different mutations. Within the cardiac space, we have a hypertrophic cardiomyopathy line, a dilated cardiomyopathy line, and then we've looked at BAG3 as well. So quite a few different disease models that I can think of right off the top of my head. And there's probably others that our customers are using as well.
Wonderful. Next question is, in the 2D cultures, do you see any differences in fusion index of wild type versus DMD? They seem quite similar, is a comment.
They are quite similar. And as I said earlier, really quantifying that fusion index is almost impossible. So counting the number of nuclei that have fused per single myotube in a 2D culture, it's impossible to untangle one myotube from another. And if you want to do that, you have to seed them in very low density. And then you have to wonder, well, are they so far apart that that's impacting the fusion of the cells? So really quantifying that number is a very painstaking process, and we haven't done that explicitly. So as I said, it's more of a qualitative measure. So by eye, they do look very similar. Both lines seem to have a very high fusion index, whatever that exact number is, and it seems to be pretty comparable. So basically, when we seed our cells in 2D, we want to make sure that we end up with a monolayer of muscle, and there aren't any patches in the dish that don't have fused myotubes in there.
Wonderful. Thank you, Shawn.
The other thing I would say quickly as a follow-up to that, so you can actually see multiple nuclei that fuse and form these muscle myotubes in 2D cultures. So even though we haven't specifically counted them to get a quantified index, we do know that we are getting 10, 20, 30, 40, 50 nuclei that fuse to form these myotubes. So it is highly multinucleated.
Okay, our next question. I was wondering if you have developed any proprietary media for 3D cardiac EMTs.
Yes, so that is exactly what we are doing as we speak. So I think our cardiac media will be ready for beta testing here very, very soon, probably within the next month or so. So we've been actively working on that for quite a while. And we're just working out, running a few final studies, looking at different lines. But yeah, that will be coming down the Curi Bio pipeline very, very soon. And you can also, if you're interested in beta testing our media, you can reach out to me or the team, and we can possibly get you on the list of testing this on your constructs.
That was a good one. Awesome. Thank you. Could you show function restoration of DMD cells upon ASO treatment?
Yes. Short answer is yes. So I can't talk specifically about these different treatments. So we have a services team that works with different customers on different projects using different cell lines and antisense oligonucleotides. That's one of the therapeutic strategies that our customer, more than one customer, is using actually. And so we know that our constructs do respond to this kind of therapeutic intervention. And while I can't talk about the specifics of that, this modality is certainly able to be used on our 3D constructs.
Awesome. Next question. Do you see satellite cells in 3D constructs?
That's another great question. And we don't have that data yet. So that's something that we just haven't stained for, and we need to. And that's certainly on the list of things to do. I can say that when we kill all of the myotubes in our 3D constructs with a drug, so we use barium chloride to kill everything, function actually recovers in our 3D tissues over time. So I don't remember the exact time scale. I think it's a few days or a week or something. So that would indicate that there's probably satellite cells in there, but we haven't specifically stained them to say for certain.
Perfect. Okay, next question. Interesting to hear the advantages of your muscle media. How about your myoblasts? What are their advantages over other commercially available cells?
Yeah, so I've been working with our customers for quite a while, different customers getting this 3D model up and running in their labs. And I'll tell you, there's a big difference in the way that different people culture their myotube or their myoblasts, excuse me. So the way they expand them, how confluent do they let them get before they passage them? Um, how many passages do they do? Do they monitor doubling times as they passage the cells? So all of those things actually affect the tissues that you make in the end. So we want to have the healthiest cell with a very nice doubling time. So as you passage your cells, your doubling times are going to slow. And eventually those cells are going to get to a point, there's a finite amount of doublings they're going to undergo. And that seems to be true for iPSC-derived myoblasts as well. And so what we've done is we've QC'd our cells and looked at over 9 passages, so over 20 something doublings. And we can see that doubling time slows over time. They still show nice fusion, but there's certainly something happening. They're either becoming quiescent, and it's hard to say exactly what will happen at the end of their life. So either they'll stop fusing, they'll become quiescent, or they'll spontaneously differentiate. Something's going to happen. So different customers will handle their cells in different ways, and they get different outcomes in the tissues that they make. Sometimes they really struggle with making functional tissues. And I have to worry that sometimes it's the way that they're growing their cells. So with our cells, they're QC'd. You thaw a vial and you're going to get functional tissues every time. That function is validated lot to lot. So we can make sure that the function between our isogenic pair is the same between every vial that you thaw, or within a range of function. And so I would say that's one of the biggest differences with the cells that you get from us is they are validated in 3D. So we know the function that you're going to get, and we know that it's you're not going to be impeded by growing cells for 3D tissues is a little different than growing them for 2D because you have to be much more careful with the way they're handled, and that you're not allowing them to basically start differentiating by becoming too confluent and giving signals to other cells to start that differentiation process, basically becoming primed for differentiation.
Hey, thank you, Shawn. Next question is, do these tissues require accompanying fibroblasts to form properly and provide longevity in culture? And then the same individual asks later, what about fluorescent imaging in the chambers? It's kind of 2 parts there.
Yeah. So when we first started optimizing our skeletal muscle platform, we were not using fibroblasts. So you don't have to use fibroblasts in the skeletal muscle constructs. I will say force, longevity, function overall is greatly improved by adding that extra cell type in there. And it's really more physiologically relevant, right? So your native muscle has fibroblasts in there. So what we found is the stability of the matrix over time is greatly benefits by having that extra stromal support cell in there. So you don't have to, but you should. As far as fluorescent imaging, I think was the question, can you fluorescently image these? So yes, you can. So the way we do have protocols optimized to take these tissues off of the posts, embed them in paraffin, section them, and then stain them. You can also take them off of the posts and stain them just using whole mounts, and then use confocal imaging to image the entire construct. As far as keeping them on the posts, I think that would be pretty difficult because they're actually raised up off of the bottom of the plate a little bit. So I think you wouldn't probably be able to image them that way.
Okay, next question is, what DMD phenotypes, like exon deletion or mutations, iPSC cells does Curi Bio offer?
Yeah, so we have our isogenic pair, and that's a deletion of exon 45, and then a 17 base pair deletion of exon 54. We have several patient lines. One of them is a deletion of exon 46 through 48, exons 46 through 48. We have a deletion of exon 51, and we have a deletion of exon 50, those 3 additional patient lines. We have another patient line that has a duplication, but I'm not sure we are able to sell that one yet. So I would have to reach out to me if that's something that you're interested in, and I'll do some more digging on that one.
Fantastic. Thank you, Shawn, so much for all those answers. And thank you to everyone for sending in all your questions. We have now reached the end of our Q&A portion. If we did not get to answer your question, which there were some we didn't get to, please do connect with us via email at contact@curibio.com. We would love to make sure those questions get answers. Big thank you to everyone for participating in today's webinar. You will be receiving a follow-up email from us with access to a recording of the event. And then, of course, another big thank you to our speaker, Dr. Shawn Luttrell, for that wonderful presentation. We hope you all found this webinar informative and have a wonderful day.
Thanks, everyone.